Hello and welcome to Chapter 8. We're going to talk about chromosomes and cell division with your friendly neighborhood professor, Dr. Andy Backey. Okay, so some learning objectives to start out with. There's actually a lot of learning objectives here. I'm only going to cover some of them, but we got to understand the different types of cell division. There's actually 3 types of cell division that we're going to learn about. When we start to get into eukaryotic cell division, focusing on mitosis and meiosis. So mitosis is all about getting rid of old cells, replacing them with new duplicates, and meiosis is all about how you make gametes, sperm and eggs. We're going to talk about sex differences in chromosomes and talk about like how sometimes cells don't divide properly and you get different or wrong numbers of chromosomes in there. So those are our learning objectives. Let's just dive right in and start talking about how cells actually age. And there's a structure on the chromosome called a telomere. A telomere is a little bit like your car's odometer, because like an odometer tells you how far your vehicle is driven, and animal cells especially, and remember, you are an animal, belong to that group of animals that have animal cells. We actually have this structure called the telomere that's very much like an odometer. Every time a cell divides, the telomere gets a little bit shorter, and eventually this protective kind of end cap that you have on the chromosomes that gets broken away. Every single time cells divide, it gets broken away, and eventually you start to get into the actual bit of chromosome that carries information, the functional part of the DNA that carries information, and that is not good. That's definitely no bueno. So telomeres are very important. We do know that when you're born, most telomeres are long enough to get maybe 80 to 90 cell divisions. By the time you turn 70, you've only got enough of about 20 to 30 cell divisions left. And so that's one of the reasons that we age, and we can talk about that more, but we are going to hit this a little bit as we get into talking about what happens if you are born with telomeres that are actually too short or much, much shorter than they ought to be. And so, there is a disorder associated with being born with telomeres that are too short. It's called Hutchinson-Gilford progeria. And progeria literally means early aging. So geriatric, you can kind of think about that. And pro meaning birth before or early. So, people with Hutchinson-Gilford progeria have these very short telomeres, and every time their cells divide, they don't have that protection, so they start to get into the main chromosome. Children who are born with progeria have an appearance of older age. You can see like wrinkles and thin skin and balding, those kinds of things. So that might actually make you start thinking about like, well, why can't we just rebuild the telomeres? I mean, that would not only help people with progeria, but it would also help people like who are actually normally aging, getting into 60s, 70s, 80s, and maybe not suffering some of the consequences of aging. So there are some cells that can rebuild their telomeres, including like single-celled organisms, eukaryotic single-celled organisms. The reason I'm focusing just on eukaryotes is because, remember, prokaryotes have circular chromosomes, so they don't have telomeres. Only eukaryotes have telomeres. So some single-celled eukaryotes rebuild their telomeres every single time they divide. But this kind of makes sense because if you think about like a paramecium, when it divides, if its telomeres get shorter, then basically eventually there'd be no more paramecium. So they rebuild their telomeres. In humans and multicellular organisms, cells that produce gametes make those telomeres. This also makes sense because if you are making a gamete that eventually might become fertilized and become a fetus, and a fetus, be born and become a baby, then you would want that baby to have full-length telomeres, unlike our progeria children. And finally, cancer cells. So cells that are cancer cells are constantly dividing, and they divide in an uncontrollable fashion, and they keep doing it, keep doing it, keep doing it. So that's bad, and that's cancer. So that's telomeres. We'll come back and revisit telomeres in a little bit, but for now, just kind of keep that in mind as we talk about next the differences between prokaryotic cells and eukaryotic cells, especially when it comes to those telomere things. So I already reminded you that prokaryotic cells have this circular chromosome, right? There's no ends, so there's no telomeres. But they both store genetic information, and they are called chromosomes. So this is a chromosome, this is a chromosome. They're both made up of DNA. And, oops, sorry about that. And they're both made up of DNA. And interestingly, they both have these kind of proteins that bind onto the DNA that help them coil up even further. And when we get a little bit into cell division process, we'll talk about how DNA coils and uncoils. I want you to keep in mind these proteins here. In eukaryotes, they're called histones, and the DNA coils up. When DNA is uncoiled, we use this term chromatin. So chromatin is an uncoiled chromosome. But the word chromosome, we still are using this word chromosome fairly loosely. In this chapter, we tend to use the word chromosome to describe not only the individual strands of DNA, but also a shape of DNA where it's all coiled up around these histones. So we've got our circular versus linear, we've got histones, and so forth. So those are the major differences between prokaryotic cells and eukaryotic cells. Of course, you got the nucleus versus no nucleus. And actually, that's a little bit important too. As we learn the difference between cell division in eukaryotes versus prokaryotes, this nucleus is going to have to be dealt with. In prokaryotic cells, the fact that the DNA is actually attached to the plasma membrane, that's going to be something that we'll deal with, and they all become important to the process. So we're going to start out with learning about the cell division that happens in prokaryotic cells, and really fairly straightforward here. So I just showed you before that the DNA of the prokaryote is actually attached to the plasma membrane at this location that your book calls the attachment site. And that's important, because as the DNA is attached to this attachment site, and then starts to go through this process of DNA replication, eventually what you get, and this DNA replication, just for now, we're just going to do really, really basic, is that we have the DNA unzipping. DNA polymerase is doing that unzipping. It comes in and it copies, puts the new bases in on both strands of the DNA. And in this circular DNA, it's kind of going along both strands until it finally, eventually gets completely copied. Once it gets copied, both strands of DNA, both double-stranded strands of DNA, circular strands, are attached to the plasma membrane. So this one's attached like right there, and this one would be attached like right there. And you can kind of see that in the cell here. So what happens next is the cell division process. Like, replication is all preparation for cell division, but cell division happens after replication happens. And so in binary fission, this is very, very basic. The cell elongates. It gets longer and longer and longer. And as it's getting longer, it's actually pulling this two strands of DNA farther and farther apart. So it's elongating and pulling the DNA apart. Eventually we get these pinching in, and we get two separate cells, each of which are called daughter cells to the original parent cell, which is up here. So these two daughter cells are genetically identical to the parent cell because the replication process would make identical copies of DNA, and so those just get pulled apart. We call this process binary fission, but we also can use the term asexual reproduction because we are making offspring, so reproduction, but sex is not involved, so asexual reproduction. Eukaryotic cell division is just very, very, very much more complicated. So I'm going to talk to you about some of the complications that are involved in the eukaryotic division process. And we're going to start out with the asexual process, which is, we just learned, is reproduction without sex. So we're not making babies here, we're just making new exact copies of cells. And when we talk about eukaryotic cell division, we do talk about something called the cell cycle. So that's a big term for you, the cell cycle. And all that the cell cycle is the process by which you can make, prepare and make new cells. So the cell cycle. We're going to start out in the cell cycle here with this step number 1. This is called gap 1. And it's called gap 1 because there's nothing really happening here in the cells with regard to the DNA. Like we're not doing anything to the DNA to get ready for cell division. So it's called gap 1. It's the first of the stages where normal cell processes are happening. Transcription, translation, all that good stuff is happening. But there's nothing like specifically happening to the DNA prepping for the mitotic phase. Sometimes cells will stay in gap 1 for a really, really long time, like years. And if it stays in there for a long time, even if it's just days, we call it gap 0. And 0 just means pause or resting. So this is the normal phase that cells will stay in for days, weeks, months, years. That's gap 1. Again, making proteins, doing normal cell stuff, that's all happening in gap 1. The second phase of this process, and all this process in purple, is called interphase. Interphase is the S phase, phase 2, the S phase, and that stands for DNA synthesis. So with the S phase, what we're doing is we're actually making copies of our DNA. Very similar to how we've already talked about it in this class. The DNA opens up, the DNA polymerase comes in and adds the new nucleotides, and that's all happening in S phase. And in other chapters, we look more closely at what's happening exactly to that DNA. But for now, just for looking at it for now, I want you to think about it, how it looks at the level of the whole nucleus. So we have our chromosomes here. This is our linear chromosome. You know it's eukaryotic because it's linear. And with replication, we get this interesting X shape. So we have this X shape where our chromosomes are actually attached together at this central part. The central part is called the centromere. So now that we have our replicated DNA, it stays in this phase stays in this formation, I guess, until the next phase is through, and that next phase is called gap 2. So inside this nucleus in gap 2, we have the replicated chromosomes. Inside the nucleus in gap 1, we have these unreplicated chromosomes, like so. So with gap 2, except for the fact that the chromosomes are now replicated, whereas in gap 1 they were not replicated, everything is really very similar. Cells are doing their normal thing, but they're also kind of doing some preparation to get ready for cell division. Some of that preparation might include things like making centrioles, which will be an important term for when we get through cell division as well. So that's gap 2. And again, gap 1 or gap 0, depending on how long it stays in that gap 1, the S phase, and gap 2, all together, that's called interphase. Now we're ready to get into the mitotic phase, and the mitotic phase includes mitosis, which is we really spend so much time focusing on mitosis, like that cell division. We have a whole lab, like half of the lab is next week is devoted to mitosis. But the mitotic phase is not just mitosis, it's also cytokinesis. And cytokinesis is how the cytoplasm divides. When we were looking at prokaryotic cells just a second ago, we really didn't have to worry about separating out cell division from cytoplasm from cell division of nucleus because there was no nucleus. With eukaryotic cells, because there is this structure called the nucleus, and that's where all that DNA is, we kind of have to separate it out. Mitosis focuses on the nucleus. Cytokinesis focuses on the cytoplasm. So just wanted to talk to you briefly before we get into the nitty-gritty of mitosis, that your cells actually do regulate. They do check themselves to make sure that they're ready to go to the next phase. And so we call this the cell cycle control system. And there are 3 checkpoints in this control system that says, okay, are we ready to do the next stage? Are we ready to do the next stage? And again, those are critical. If any cell is not ready to go to the next stage, then it'll literally put the kibosh on it. You will just stop where you are. The checkpoints really make it possible to reduce the likelihood of cells having errors in them, or if they have errors that they won't go ahead and finish the cell division process. It allows cells to kind of figure out what's going on in their cell's environment, like the environment of your whole body, and so that's really important. So let's take a look at these checkpoints. Our first checkpoint is at the end of G1, before S starts. And if you think about it, if you think about what's happening at the end of G1 or before S starts, we want to make sure the DNA is not damaged. And we want to make sure that the cells have enough nutrients in order for that cell to keep growing and get big enough and have enough proteins in order for us to go through the very complicated stage of DNA synthesis. So if everything's good, if we've got enough nutrients, if we've got a big enough size and so forth, then we're going to go ahead and go past this checkpoint and go into the S phase. Now remember what the S phase was all about, replicating DNA. And so at the end of the G2, we make sure that S phase has completed properly. We want to make sure that all the chromosomes are replicated and that DNA synthesis happened in the way that it needed to. Did we make exact copies and so forth? So that's the G2M, and M stands for mitosis in here. So the next checkpoint is going to be during the process of mitosis itself. And specifically here, and this is not a term that I've used yet with you, but specifically what we're looking for is something called the spindle assembly. And I will be using that term for you when we get through and start talking about mitosis. But basically, the spindles are fibers that attach to the chromosomes and actually pull those chromosomes apart and separate them. So we want to make sure that those spindles have attached properly to the chromosomes and so forth. If that checkpoint gets passed, then we actually can go ahead and complete our cell division process. So just to kind of summarize what I've said so far is eukaryotic cells, specifically the ones that we're calling somatic cells, and somatic cells are body cells basically, anything except for sperm and eggs. So somatic cells and eukaryotes spend some time in the cycle between cell division and the normal cell activity is called interphase. The only part that's actually the cell division, that's the mitotic phase. And then of course, the rest of the time it's called interphase. Interphase itself has 2 separate gap phases, which are separated by the S phase or the DNA synthesis phase. And then finally, we do have checkpoints that will stop cell cycle from progressing and maybe even end up killing a cell if it's not progressed properly. So in prokaryotic cells, we learned that before the cell could divide, we needed to have chromosome replication. And in eukaryotic cell division, we have the same exact thing. Your book goes into a lot of detail about how this happens. And I am not going to go into that level of detail, nor am I going to expect you to know that level of detail that your book goes into about how chromosomes make copies of themselves. Here's what I want you to know, just the basics. The parent DNA molecule, that's the original DNA molecule. That parent molecule unzips because of DNA polymerase. Yes, you still have to know DNA polymerase. Again, not talking about the details here, but knowing that DNA polymerase adds the new complementary nucleotides, like we've already talked about, and they do go in opposite directions. But eventually what you end up with is two identical daughter DNA molecules. And just so you know, when it happens, those daughter DNA molecules are actually attached at that centromere when they are made. If the same process happens in a prokaryote, those daughter DNA molecules are attached not to each other, but to that attachment site. Hopefully that makes sense to you. Again, in a eukaryote, they'd be attached to each other at the centromere, and in a prokaryote, they'd be attached at the attachment site on the plasma membrane. So a lot of times when cells are going through cell division, they actually make mistakes. Like sometimes instead of matching A up with T, sometimes A gets matched up with C or G. And that is a basically normal process. Like errors happen. And those errors are called mutations. Like we've talked about mutations before, but really haven't thought about it in the terms of like exactly what that mutation is. And so mutations, the spontaneous kind of mutations that I talked to you about in a previous chapter, that can happen just because DNA polymerase is putting the wrong nucleotide in a place and where it doesn't belong. That's how that mutation happens. It can also happen because of like chemicals and radiation, which we've already talked about as well. But here focusing just on the job of DNA polymerase. So mutations do happen, and most mutations are not good. But I mean, we can understand that. But sometimes errors are a good thing. And I thought maybe I'd just leave this question in here, like to think about, well, why would a mutation be a good thing? Could it be potentially good? And the only way to understand how new genes, because a mutation that works well is a good mutation, the only way that this works well is if the mutation happens in a sperm or an egg. So the mutation has to kind of be there from the very, very beginning of the conception of that new organism. And we're actually going to explore the relationship between mutation and what could happen if a good mutation happens in the chapter that's coming up on evolution. So that stand by on that. We're really basically mutations can be good if it helps people survive, if it helps organisms survive and reproduce. And again, we talk about that more in the evolution chapter. So we do know for sure, getting back to cell division here, we do know for sure that mitosis generates new cells. And those cells are as best as they can be exact copies of the original cells. So why do we go through this mitosis? There's basically two reasons. Either an organism is growing, and the way that you get growth is by having more cells, and more cells means mitosis. So this is like the tip of an onion root that you might have seen in lab. We can also go through mitosis by if you end up killing a cell, and sometimes, you know, when you shave, you cut your skin, and that kills those cells, and they have to be replaced. So replacing damaged structures is also another important thing for mitosis. I like this question up here, what is dust? Because basically dust is dead skin cells. I mean, if you were to analyze dust, it's dead skin cells. So you, every day, you slough millions of dead skin cells, you and the people around you. And yet your skin, it doesn't disappear. And again, that's because you're just replacing those skin cells that died due to them being sloughed off. So yeah, well, that's gross about dust, but it's interesting about that your body has this ability to replace worn out cells. So almost every single somatic cell, and remember, definition, somatic cells are your body cells. They're everything except your eggs and sperm. Almost every somatic cell undergoes mitosis. And these things happens at different rates. So red blood cells, you're going to get new red blood cells every 2 to 4 months. You'll have a complete turnover of red blood cells. But the cells lining your intestines, they're exposed to all kinds of roughness. They're exposed to all sorts of maybe toxins. And so they're replaced much, much more frequently, every 3 weeks or so. So replacing worn out cells. Sometimes cells will actually, and this sounds a little weird, but they'll commit cell suicide, meaning that they will die and then have to be replaced. One example of this that we could talk about is when you were a fetus inside the womb, your fingers had webbing between them. And now your fingers don't have webbing, maybe just a tiny, tiny little bit at the base of your fingers, between your two fingers. But it used to be that when you were a fetus, that webbing went all the way up almost to the tips of your fingers. So we say that those cells that made up the webs went through apoptosis or cell suicide in order for you to not have that webbing. Let's face it, I mean, webbing might be cool, but as land animals, we really don't need that. The other thing that I wanted to talk to you about is when we talk about different rates of cell replacement, that some cells hardly divide at all. For example, your heart muscle cells or the neurons in your brain, they divide very, very slowly, and we're not sure why it's so, but if you have a heart attack, those heart cells die because they're not getting oxygen and they just don't replace themselves. So that damage is basically permanent. Same thing for brain damage. So we're at this time, I think we're ready to dive into talking about how mitosis actually happens. And I'm going to start that out in the next video because I've been doing this with you for almost a half an hour. So we'll take up in part 2 in just a moment.